Fresh leafy green vegetables, or LGVs, are more vulnerable to contamination by pathogenic bacteria while growing in fields. The consumption of fresh LGVs associated with a number of foodborne disease outbreaks, raising concerns about their safety. It becomes clear that relying solely on conventional sanitizers for post-harvest treatment cannot provide complete food safety protection for consumers. In-field sanitation of LGVs could provide additional levels of food safety protection. Electrolysed water has gained interest as a sanitizer over its toxical counterparts in the food industry, owing to its broad-spectrum antibacterial activities, ability to manufacture on-site cheaply, and low environmental impact. In this project, we'd like to determine the most effective sanitizers in killing Listeria inocua on LGVs. The reason to study Listeria is that Listeria inocua is a non-pathogenic Listeria species found in similar environments to Listeria monocytogenes. Listeria monocytogenes are widely spread in agricultural environments. Vegetables may be contaminated through soil, water, and the use of manure as a fertilizer. It causes potentially fatal foodborne infections in humans with a high mortality rates. To examine the efficacy of pre-harvest use of different sanitizers, 7.5 log Listeria inocua was sprayed on 4 different LGVs, using 20 ppm electrolyzed water, 20 ppm nylate, and 80 ppm tsunami on farm to inactivate the bacteria cells. On Mizuna, tsunami on farm was the most effective sanitizer against the Listeria inocua. Similar results were also observed on red chard, rocket, and spinach. Nylate solution were found to be less effective against the Listeria on 4 LGVs, probably due to the low pH of the initial nylate solution, where a fraction of active ingredient is in inactive form. Electrolyzed water were also very effective in killing Listeria inocua, using 4 times lower concentrations than tsunami on farm. To determine the impact of product quality and shelf life after sanitizer treatments, those non-Listeria inoculated leaves were stored at 4 degrees for maximum 12 days. The total bacteria populations were significantly suppressed during the first 5 days of storage. After Mizuna treated with tsunami on farm and electrolyzed water, red chard treated with electrolyzed water in comparison with water. On spinach, the total bacteria populations were significantly suppressed by tsunami on farm for 7 days in comparison with water. However, no significant difference between water-treated and sanitizer-treated rocket throughout the storage period. The suppression of total bacteria populations on Mizuna, red chard, and spinach may be implications for improved shelf life. For the sensory attributes, no changes in color, defect, texture, odor, pH, and moisture were observed during storage period across all treatments on 4 LGVs. In conclusion, tsunami on farm was the most effective sanitizer against the Listeria inocua, but it requires a high cost and high concentration to achieve the good reductions in microcounts. Electrolyzed water as a safe, cost-effective, environmentally friendly sanitizer can be used as an alternative to tsunami on farm and other conventional sanitizers. The use of pre-harvest sanitizers can significantly reduce the populations of Listeria inocua in comparison with water, and also suppress the growth of total bacteria populations during the storage period. Conceivably, pre-harvest application would be improved provision of safe products that may not require extensive post-harvest processing, but also may achieve other benefits, including increased product stability and extended shelf life.